You're muted. Can you hear me?
Yes, I can hear you. Thank you.
Okay.
How many contestants do we have today?
I think as of now we have 3. I don't think there's going to be any more. So it should be pretty fast.
thesis competition. We have 3 outstanding judges who I would like to introduce. First, Dr. Paul Luebbers from HPER. Also, Dr. Carol Lucy from the School of Business. And as well, Dr. Charles Brown from Social Sciences. Thank you to the judges. I'd like to spend a little time before we get started going over the rules and talking about judges' expectations. So let me share screen here. So the rules for the 3-minute thesis competition: a single static PowerPoint slide is permitted, and I believe Sandy has the ability to allow you to share screen at your time. Let me ask that you have no slide transitions, animations, or movement of any type. No additional electronic media, that is sound or video files, are permitted. No props, no costumes, musical instruments, lab equipment are permitted. Presentations are limited to 3 minutes. I am the timekeeper. I will give you the starting signal, as well as give you a brief 30-second warning by saying 30 seconds when your time is almost up. We will stop you at a complete 3 minutes. Presentations must be spoken word. That is, no raps, songs, or poetry. And the decisions of the judging panel is final. Judges will be looking at your communication style. So was the thesis, dissertation, purpose, and significance communicated in appropriate language for a non-specialist audience? Also looking at comprehension. Could the non-specialist understand the research? As well as engagement. Did presentation make the audience want to know more about the topic? And of course, as you probably all know, there are 3 prizes up for grabs. The winner will receive a $500 stipend, the runner-up $350, and the 3rd place contestant will receive $250. So those are the rules and what the judges will be looking for. So I understand that we are going in random order, basically, and Sandy will let you know as you are being queued. So after your presentation, the judges will make decisions based on the scoring sheets that they have been given. They might have some brief time to talk, so we will have a little bit of time between presentations. But basically, we will be going forward. I understand that Stephany is first.
So hi everyone, my name is Stephany Graham and I'm studying clinical psychology here at Emporia State University. And my thesis is over the effects of self-efficacy and motivation on fluid intelligence as measured by the block design subtest of the Wechsler Adult Intelligence Scale. And so I briefly want to go over the block design subtest just to give you an idea of what my experiment looked like. And so here's a picture of the block design. And so this subtest measures spatial reasoning, fine motor skills, as well as assesses fluid intelligence. And so each participant is given 9 cubes, and each cube has either solid red, solid white, or half red, half white on each of the sides of the cubes. And you are asked to recreate a picture using those cubes. And so my experiment had 3 groups. My first group is a control group, so there were no manipulations to this group. The administrators were told to implement the block design as the manual suggests. My next group is experimental group 1, and this group is designed to increase self-efficacy. And so how this was done was through positive affirmations. So each administrator gave positive affirmations to each participant throughout the block design subtest, and this was thought to socially persuade each participant in thinking that they were performing well and thus increasing self-efficacy. My next group is experimental group 2, and this group was designed to increase motivation. And so we did this through financial incentives, stating the time limits and value of each item. And so essentially the administrators told each participant you have, you can, if you earn a score of 55 or greater, then you can receive a gift card. And then throughout the subtest, they told them how long they have to answer each question, as well as the value of points of each item. And so to analyze my groups, I used an ANOVA. And so essentially what all this is saying is that I didn't receive statistical significance between my 3 groups. However, if you look at my eta squared, which is right here, I have medium effect size. So what that's telling me is that I have that something is going on here, and it's likely that my smaller sample size is what impacted the significance of the study. And so unfortunately, because of COVID-19, I was unable to get as many participants as what I would like. And so that might have impacted the statistical significance of my study. However, I think that this research is very important, even though I didn't get significance, because there's really no research out there on how self-efficacy or motivation impacts test performance on intelligence tests. And so I also think that this could be applied to academics as well as psychometrics. And so even though I didn't get significance today, I think that we can state that self-efficacy and motivation might not have as big of an impact on test performance like we might initially theorize. So I think that's probably all the time I have for today, and thank you so much for listening.
Thank you, Stephany. Very well done.
Thank you.
Okay, judges, if you would go ahead and score your scoring sheets and
Okay.
Okay, Bethany, whenever you're ready.
Okay. So my name is Bethany Roberton. I'm a master's student in the biology department. My research is looking at the effects of season of burn treatment on plants and pollinators in a prairie. So I chose this research because prairie habitat is very important and because it's been in decline in the past several years. I'm looking at different season of burn because springtime burns are the most commonly used, and there's a lot of negative effects with spring burns. There's not a lot known on how these burns affect pollinators. So my research took place at the Ross Natural History Reservation. So that's the picture that I'm showing you guys. I just think it's nice to look at it visually. So we have 18 experimental plots near Americus, Kansas, owned by ESU. And so I'm using these 3 summer burns that are outlined in black, the 3 fall burned plots outlined in white, and the 3 that are outlined in yellow are spring. So my research was looking at these 3 main goals, which were how do these different season of burns affect plant diversity? How do they affect a plant's ability to attract pollinators? And also, how do they affect plants' reproductive success? So to do that, I used milkweed species. So I had different milkweed species, 4 different ones out here at the Ross. So I was looking at these different plots, doing plant surveys, doing nectar collections with sucrose concentration, and also doing pollinator surveys, biomass, seed viability assays, all using these plots. I did my first season last summer, and I have my second one coming up this summer, and we're actually going to be using all 18 plots instead of just 9 this upcoming summer. So with my different methods of data collection, I was addressing those 3 main goals that I had, like I said, looking at diversity, looking at the plant reproductive success, and a plant's ability to attract pollinators. So overall, I found that summer burns had the highest plant diversity, which was great. So that conservation implication is that summer burns are probably the best in the short term for prairie restoration sites because they promote plant diversity. However, I did not find any difference in the nectar sucrose
30 seconds.
concentration or in seed viability for between these plots. So that indicates that even though plant diversity was affected by season, the actual reproductive success and ability to attract pollinators was not affected. So that can also be very important for conservation and restoring prairie sites. So with that, I just want to say thank you for listening to me today, and that is it.
Thank you, Bethany. Whenever
Okay.
you're ready, Jim.
Okay, I'm all set. I'll begin now. Hello, everyone. My name is James Miazga, and today I'll be describing my research I'm working on for my master's thesis involving crappie management in Kansas. So why are we interested in managing crappie populations? Well, they are a popular sport fish that are highly desirable among anglers because they are abundant, relatively easy to catch, and they're highly preferred for consumption. Additionally, crappie fisheries are economically valuable. In 2011, crappie anglers spent $210 million on fishing-related expenses. So clearly there's a need for quality crappie fisheries, and understanding these fisheries is important. We have limited understanding of the population dynamics of crappies in Kansas. Dynamics of fish populations are rates of recruitment, growth, and mortality, and these are important for making management actions. Harvest regulations are effective management tools at managing crappie populations. They reduce angler harvest. However, in some cases, this can produce undesirable population level effects. Because we have a limited understanding of the population dynamics, we're not sure how effective our current harvest regulations are for managing crappie populations. Therefore, my objectives are to describe population dynamics and evaluate our current harvest regulations. This begins with age estimation of individual fish within a population. Age can be estimated from otoliths, as seen in the bottom picture. Otoliths are inner ear bones of a fish, and annuli, or these growth rings, can be enumerated to determine an individual's age. This age data is then compiled and used to calculate population dynamic rate functions. These dynamic rate functions are incorporated into harvest models that are used to predict angler harvest in kilograms. We can use these models to determine which harvest regulations produce the most angler harvest. Upon completion of this research, we will have compiled baseline level population information that was previously unknown and use this information to inform statewide harvest regulations to improve crappie management in Kansas. I'd like to thank the Kansas Department of Wildlife, Parks, and Tourism for funding the project, as well as my advisor, Dr. Zach Klein. And that's all I have.
Thank you, James. Appreciate your presentation.
Thank you.
Okay, so we have Isaiah Wattree. Isaiah, are you ready to begin?
Yes.
Okay, as you begin, I will start the clock.
Okay. So when it comes to work, I always find myself asking these two questions. Do I have to go into work today? Or can I work from home? Now, if any of you have ever asked similar questions, there's good news. You can definitely work from home. More and more organizations are allowing their employees to pick when and where they do their work. These options are known as flexible work arrangements. Previous research tells us that when an employee has flexible work options, they're able to spend more time with their families and respond to emergencies when needed. This leads to a better home life. In return, that employee will feel more satisfied with their job. In my research, I wanted to learn about the positive benefits of flexible work arrangements. I wanted to see how flexible work arrangements influence burnout and an employee's commitment to their organization. To test this, I surveyed 202 participants, some with traditional schedules and others with more flexible schedules. I wanted to see how these participants felt about their lives and whether or not they felt committed to the places they worked. I wanted to see if they felt burned out as well from the work that they did. What I found was that participants with more flexible schedules also reported having a better home life, being more committed to their organizations, and feeling less symptoms of burnout. Another interesting finding from my research is that flexible work arrangements are not directly related to burnout, nor are they directly related to employee commitment. It's only when I asked about home life that I saw significant relationships. This shows how valuable home life is to an employee. These findings are important because times are changing and technology allows us to do work whenever and wherever. The situation facing our world now is the greatest example of this. COVID-19 has opened the door up for many positions to be done remotely, and when this is over, there's a strong possibility that work will never be the same. As more companies begin to change how work is done, it is important for us to continue to research the effects. Companies who choose to stay with traditional schedules are only going to be left behind by their employees and by society itself. Thank you.
Thank you, Isaiah.
Thank you.
So I can share it now.
Okay, whenever you're ready, Shelby.
All right, I'll go ahead and go. My name is Shelby Young, and I'm a graduate student in the Forensic Science program, and I chose to do my research over forensic soil analysis. And I did this because right now we're experiencing a lack of using soil evidence except for high-profile crimes, and this is especially true for Kansas. And this is because soil evidence, just analysis in general, is complex, it's time-consuming, the instrumentation destroys the sample, and it's expensive and requires an expert. So this is why we don't really do it in Kansas. And I wanted to find an instrument that almost every forensic laboratory has. And I did find one. And if you look over on my slide, it's called Attenuated Total Reflectance Fourier Transform Infrared Spectroscopy. And that's really a mouthful, so I just call it ATR. It's a whole lot easier. So the middle image is a picture of the ATR. And it's really just plain looking. It's pretty small. It's called a benchtop instrument. And it requires a very small amount of sample, which is a benefit in forensic science because you never really know what kind of evidence you're going to get. So I put soil on the little circle. I don't know if you can see my mouse, but I just put like a couple milligrams right there on that circle. And I pull the handle down and it takes a couple scans and I get a spectra similar to the one on the right. So what I did when I got the samples, I went out to the Ross Natural History Reservation, and that's in the left image, and I collected 100 different soil cores. So it took many days because that's a lot of soil to collect. And I chose 5 diverse locations. So basically they had different soil compositions. And I took this soil, I split it in half because I wanted to see if there was also a difference between the layers of soil. And then I dried and I blended it for homogenization just so I could get a representative sample. And then I used the ATR and I scanned each sample 5 times. So overall, I had over 1,000 different spectra to kind of sift through. And I only have one example here on this slide. And that picture there has over 200 spectra stacked on it. So that's just one area. And at first I didn't see a whole lot of differences between the different areas, but spectra is like, it's not very easy to see to the human eye. So I used something called principal component analysis, and this is just a multivariate technique that helps you see those minor differences that you can't see as easily to the human eye. And with this method, I was able to get 100% identification of soils because I also used blind samples to test the theory, and it worked. So overall, ATR is an efficient technique to use as a preliminary method for forensic soil analysis. So I just hope it gets used in the future.
Thank you, Shelby.
Mm-hmm.
Okay, so the title of my thesis is Losing Our Humanity, the Lovecraftian Sublime and its Paralysis of Anthropocentrism. And I think the title captures most of what's there. So what I was researching for my thesis is the fiction and nonfiction of H.P. Lovecraft, who was an early 20th century author of cosmic horror, who mainly contributed to the pulp magazines. And cosmic horror differs from your ordinary brand of horror because it doesn't necessarily study zombies or vampires or any other such things. It's more about a feeling of existential worthlessness. So what often happens in a work of H.P. Lovecraft's is a human observer or subject has an encounter with the vast or the infinite reaches of space, and it induces in them a sense of paralysis, and freezing oftentimes leads to madness or death or some kind of transformation. But the general philosophical idea that underlies H.P. Lovecraft's work is that humans are trivial, that there is no purpose, no meaning, no value. So I think the no purpose, no meaning, no value captures Lovecraft, but it also captures more generally a sense of nihilism. And that nihilism is often perceived by literary critics as a critique of humanism, or more specifically of anthropocentrism, which is the view that humans are essentially the center of everything, everything revolves around us, and so forth. So this, I believe, is the Lovecraftian sublime, as I call it. That encounter with the vast or the infinite paralyzes reason. And it differs from the humanist models of the sublime because in the humanist models, humanity prevails. Human reason can conquer all, it can overcome nature, and that's what's truly sublime, is the human. That's not the case in Lovecraft. So that divergence from humanism is viewed by post-humanist critics and eco-critics as the critique of humanism. And Lovecraft's materialist philosophy breaks down the subject and object dichotomy. And so these are all seen as a kind of common ground with H.P. Lovecraft. And it's often viewed also as Lovecraft being a kind of precursor to post-humanism and eco-critical ideas. However, I think where some critics ought to be a little more cautious is in their suggestion that this materialist, nihilist base can function as a jumping off point for more ethical modes of being. I don't believe that H.P. Lovecraft leaves any room for morals or lessons. He's unlike other authors in that his work can't really offer any advice or course of action, because what would be the point of a course of action or a moral in a universe where we don't matter and we're temporary and trivial? Anyway. Is that time?
Yes.
All right. Thank you.
Thank you, Nick. Really appreciate it.
Okay.
Thank you all for joining this year's 3-minute thesis competition. We hope to be live and in person again next year. And we will make a public announcement of the winners very shortly through the announcement system. So thank you all for joining. And thank you to the judges again, Dr. Luebbers, Dr. Brown, and Dr. Lucy for their participation.